Psst, I see small GTPases. A transport on that beat, yo. Small GTPases hydraulic GTP, converting it from triforming to GDP. Involving translocation, vesicles, and nuclear, we're gonna zoom in on the nuclear pore. Start off with a cargo that has an NLS, nuclear localization sequence, yes. The protein binds one factor called the important, gets cargo in the nucleus, very important. Once the duo is in, it binds to GTP. It breaks off the cargo in the nucleus, it's free. Important GTP complex exits through the pore, along the concentration gradient, that's what it's there for. Rand gap binds the complex, and hydrolysis Rand, all three dissociate, it's almost like when it began. One more thing to do, when GDP goes down, it's CG into nucleus, making the way back around. GEF in the nucleus changes from D to T, and the cycle starts again, because everything's ready. Next, I'm going to talk about nuclear export. Our goal is to get the cargo from the nucleus out the pore. From export of any S cargo, we need three factors in nucleus, binary cargo, and GDP, and export 10. The trio exits through the pore and binds Rand gap, converts T to D, and the three unsnap. It leaves with the GDP exports and a cargo. In the cytos or real quick, it doesn't take largo. GDP and E rebinding complex crosses the nuclear membrane to restart the process. Back inside, they unbind and Rand gap, phosphorylates Rand GDP like what the F. The biggest process that makes this whole thing run is the concentration gradient and localization. There's a higher concentration that Rand GTP inside, so it wants to move out and brings it further right. On the outside, there's more Rand GDP maintained by Rand gap. This stuff is easy. GTP inside is maintained by Rand gap. It's a phosphorylator. Let me take a breath. Endoplasmic reticulum. Protein processing after the ribosome is post-translational translocation, but you can also have co-translational translocation. For co, you have the signal recognition particle, which binds to the newly forming peptide end reticle. This process targets new peptides through the ER, and they start the translocation process kind of bizarre. Sec61 accepts nascent protein chains, the receptor right there on ER membranes. Proteins destined for ER might have ERSS, if not they have one extra start sequence. Translocation is mediated by hydrophobic zones. Order matter is not sequenced, they could be clones. Start and stop regions are non-polar because they stick in the membrane of ER. Next big player in translocation is the heat shock protein or the chaperone. Chaperones push proteins to their folded state to avoid an amyloid and misfolded aggregate. They also help with end-link glycosylation, adding sugars to proteins to guide their location. Sometimes things are wrong, misfolding goes too far, those proteins have got to get out of the ER. The ERAD pathway, or retrotranslocation, uses ubiquitin to promote protein degradation. That's it for the ER, but we'll be right back for more. The secretory pathway is quite the endeavor. Let's go over the secretory pathway. Golgi, plasma NOA. Vesicle formation is pretty complicated, but it helps to know it's receptor mediated. Anterograde trafficking starts at the ER with a COP2 coat and a GTP is called SAR. Co-proteins assemble and give the vesicle a coat and stay until the target. Yeah, that's dope. SAR1 is activated with GTP binding with the donor. Vesicles buzz adios, donor, hardly know her. Then it's off to the Golgi, the target membrane, and it binds releases cargo, got transport on the brain. Retrograde trafficking is returning to ER. The GTPases are like a dog or retriever. Why are proteins retrograde? It doesn't make sense. KDEL sequence for ER residents. The co-proteins got blend and the pattern is the same. It's just going over to the opposite membrane. Interactions with the plasma NO side use a coat, protein, coat, and clathrin for the right. Endo and exocytosis alike use clathrin and no small GTPase, alright? And vesicles are coded, but how they get around. Certified donor membrane, certified target membrane, rab, targeting molecule, rab, binds to the protein. GTPase goes right back to donor, and you tired, trying to track the vesicle, yeah, that's probably a T-snare. For questions on experiments with mutations, you have to figure out the protein localization. More often than not, the answer is use GFP tag, but if it's not, there's another trick in their bag. A digestive enzyme from bacteria called EndoH cleaves sugars off proteins as controlled by pH. Proteins are sensitive from ER to Golgi and resistant beyond the trans-Golgi that rhymes with Golgi. An additional pathway is direction to lysosomes, M6B tagging proteins to endosomes. Last, let's talk about the gaps and GEFs to avoid any confusion or missteps. At the nucleus, Rand gap is inside. Rand gap is cytosolic. Let's go for a ride. SAR1 gap is on the ER, and SAR gap is part of vesicle co-inner. ARF1 gap is on the Golgi, and gap is on the vesicle. Stay in control, G. Now RAB gap is on the donor membrane, but gap is on the target. Its location stays the same. RAB itself moves with the vesicle, and RAB GBI is there to recycle. KDEL's at the Golgi and retrograde. M6P echo G and going lysosomes way. Endoplasmic reticulum. Nuclear. Secretory. Are we translocating? Are we trafficking? Endoplasmic reticulum. Nuclear. Secretory.